study chemical engineering at University of Michigan. My research is about isolation and analysis of circulating tumor cells. Cancer is the second leading cause of death in the United States. Although many drugs and therapies have been developed for cancer treatment, it is still quite difficult to decide which treatment works best for a particular cancer patient, because every cancer patient is somehow different. Right now, there are few efficient ways to predict or monitor patients' response to a certain therapy. Traditional ways include CT scanning and biopsy. CT scanning requires expensive and huge equipment, and in many cases, CT scanning could not reflect the changes of patient conditions timely. Biopsy is a procedure to remove a piece of tissue from the patient's body. Biopsy could be complicated. For example, in many cases, you need to use CT scanning to locate the tumor, and then you need to put a big needle inside the patient's body to scratch a piece of tissue off. Biopsy actually have high risk of bringing infections to patients or causing damage to the normal tissue of patients. So we are trying to develop a simple, easy, safe, and relatively cheap way to monitor the patient's condition. So we're thinking, what if a blood test would solve this problem? If we could just take a few ml blood from the vein of patient's arm, and then we could analyze those blood to give us enough information to reflect the patient's status. That would be fantastic. So we are trying to think, is there something special in the blood of cancer patients that could give us all those information that we need? Actually, there is. They are circulating tumor cells, or CTCs. CTCs are those cancer cells that moves around. CTCs would leave the original tumor site and enter the blood circulation. They would travel through the blood vessels and might locate in another part of the body and develop a new tumor. We could actually take advantage of those CTCs. If we could isolate and analyze the CTCs from the blood of cancer patients, we could obtain those informations from these cancer cells, and these informations could be useful to help physicians know the condition of these patients and to help him to develop personalized medicine for this cancer patient. This idea sounds simple, but actually isolating CTCs from patients' blood is a challenging work. Actually, in 1 ml of blood, there are 5 billion red blood cells and 7 million white blood cells. Well, only 1 to 100 CTCs normally. So try to isolate CTCs from all those blood cells are really like finding needle in a haystack. We need to think about some smart ways to accomplish that. So we're trying to think what's the huge difference between CTCs and blood cells. One of them is surface proteins. In every cell of our body, they have different proteins on the cell surface. Compared to blood cells, CTCs have this unique protein called EpCAM. So our strategy is to coat EpCAM antibody on a substrate. So those EpCAM antibody will only binding to EpCAM protein. They will not binding to any other proteins. They will only binding to the EpCAM protein on the surface of CTCs. So those CTCs will be immobilized on the surface of the substrate, while the blood cell will just flow through without binding to the substrate. In order to improve the capture efficiency, we use a microchip and we coated nanomaterial called graphene oxide onto the surface of the chip. So you can see here is an example of our device. This is pretty small, right? Looks like. Actually, the height of the inner chamber of this device is only 50 micrometer, so that it could create a very stable fluidic profile that could facilitate the capture of CTCs. On the surface of this device, we actually coated tens of thousands of gold patterns, and graphene oxide could be coated on those gold patterns, and in this way we could improve the specific binding of the CTCs and also improve the binding efficiency of the CTCs. The fabrication of this graphene oxide chip could be described as below. So first we have the silicon substrate, and then we coated the gold pattern on top of it, and then graphene oxide will absorb to the gold pattern. And then through some biochemistry and cross linkers, we would be able to coat the antibody on the graphene oxide with a really high concentration. And then when we flow the blood through this device, CTCs would be captured on the surface of this device, while all those blood cells will just flow through without being captured. Now we have this device that works pretty well for capture CTC, and we are currently working on testing patient samples onto this device. When the patient walks to the clinic, the physicians and the patient always have those questions, such as which drugs works best for this particular patient. After using this drug for a certain time, is this drug effective? Or along with chemotherapy, should we also use radiation therapy or not? And many other similar questions. We want to help them to answer those questions with more evidence and confidence. So what we're doing now is we take a few ml blood from the patients, and then we run through this blood to our graphene oxide chip. And the CTCs would be captured within this chip. Then we are ready to do CTC characterization. We could either staining the CTCs with different protein markers to count how many CTCs are there, what kind of protein do they express. We could also analyze the genes of those CTCs, and then we could tell what kind of genes are upregulated or downregulated within this patient, what kind of mutations could these cancer cells carry. And then we try to correlate this CTC informations we have with the clinical information of this patient, such as what kind of treatment this patient receive, and does this patient respond to the treatment or not, is the tumor shrink or the tumor size increase. So we're trying to build a correlation between the CTC informations and the patient status, so that in the future the physicians could use those CTC information to decide what kind of treatment this patient could use, and to select the personalized treatment for the cancer patients. So to summarize, those are the works that I have been doing on the benches in my lab. But my real passion is to bring our technology from the bench side in the lab to the bedside of cancer patients, so that our technology could really be applied in clinical setting and to bring the real benefits for cancer patients. Thank you.